So, hello students. This is the presentation on WBC cells and why we are so much concerned about WBC cells. That this is part of our immune system and it warns our body that any foreign substance has invaded our uh, body and WBC needs to act. So, WBC is very useful indicator of any infection in the body. This presentation looks like a big one. I would be going through um, most of the basic concepts, and for the purpose of your exam, I won't be going more deep into the subcategory of these headings, but the basic concepts, the basic components of white blood cells might get tested. So if you have leukocytes, then leukocytes, they are granulocytic leukocytes or agranulocytic leukocytes, and then in those you have eosinophils, neutrophils, basophils, and then you have monocytes, etc. So those are the concepts which I might test you on in this exam, and the rest of it might become part of your um, final exam. So let's begin. A basic knowledge of hematology is important for us, as you all know, and it evaluates the patient's ability to transport oxygen and carbon dioxide, and also hematology tells you about how intravascular volume inside the body is maintained. Hematology also takes part in coagulation of blood, and it also combats infections. Now, the hematology to define is study of blood and blood forming tissues, and we will be discussing uh, uh, some things like bone marrow, blood, spleen, and lymph system. Assessment of the hematological system is based on the patient's health history, physical examination, and the results of his diagnostic studies. Let's go to the next slide. This is telling you how uh, the purpose hematology is that it is uh, hematology does hematopoiesis, which is blood cell production, and this happens within the bone marrow. And bone marrow produces RBCs, WBCs, and platelets. And the three types of cells they develop from the immature blood cells in the bone marrow, which are called as stem cells. The three types of in this there is a blood cell development diagram here, and the three types of blood cells are RBCs, WBCs, and platelets, and they develop from a common cell within the bone marrow. The hematopoietic stem cell is actually an immature, it's not mature, it's an immature blood cell that is able to self-renew itself and then differentiate into a hematopoietic cell. So the bone marrow is stimulated by various factors or cytokines that cause differentiation of the stem cells into one of the three types of blood cells. So the root is stem cells, and then from there three types of cells, RBCs, WBCs, and platelets, they develop. What is a bone marrow? Bone marrow is a soft material that fills the central core of bones, and there are two types of bone marrow. That is adipose, yellow, so that is the where the lipid, which reflects the lipids, the fat, and the other one is hematopoietic bone marrow, which is red. So the yellow bone marrow has adipose in it, and the red bone marrow is the is hematopoietic. Bone marrow. The hematopoiesis, or the red blood cell production, it happens within the red bone marrow. In adults, the red marrow is found uh, primarily in the flat and irregular bones of your body. And some of these flat and irregular bones are the ends of long bones, pelvic bones, vertebrae, sacrum, sternum, ribs, flat cranial bones, and scapula. The marrow is able to respond to increased demand for various types of blood cells by increasing production, and they by a negative feedback system. So we don't want to go deep into the negative feedback system. It's just remember that red bone marrow is the place where the hematopoiesis happens, and the bone marrow is stimulated by various factors actually, and example, erythropoietin affects the bone marrow stimulation. Let's go to the next slide. Blood and the structure and function, the same diagram again. And the blood, when you're talking about hematological system, we need to talk about blood. And what is blood? Actually, a blood is a type of connective tissue that performs three major functions: transportation, regulation, and protection. There are about this is what I was talking, um, referring to earlier, and this is um, you see is that there is a total body weight. I'll be discussing this in the class. There is a total body weight, and out of our total body weight, the 8% of that weight is blood, and other fluids and tissues are 92%. Whole blood, the percentage by volume is that 55% of the whole blood is plasma, and the 45% is the one which is composed of RBCs, WBCs, and platelets. And plasma is composed primarily of water, but it also has in its proteins, electrolytes, gases, nutrients, and waste. And the term serum, it refers to plasma without its clotting factors. Most plasma proteins are produced by the liver, except for immunoglobulins, which are produced by plasma cells. Um, Again, they have also shown you division of plasma percentage by weight, where you have protein, water, and other substances. Again, they have shown you in protein, you have further subdivisions of albumin, etc. So this is how uh, when they are when there is a blood components, or where then how the blood components are there by percentages shown in this diagram. And when it says formed, the 45% is where you have RBCs, WC, and platelets. Is showing you how much percentage you have RBC, and how much percentage you have platelets, and how much percentage you have leukocytes, WBC, and how much percentage out of the leukocytes you have the other subdivisions of neutrophils, monocytes, etc. So this is what you should need to know. So anytime there is any change in the blood component, think about the kind of substances which will have a decrease in the body. So next, go to the next slide. This is a repetition. So plasma constitutes 55% of blood, and is composed primarily of water, but also contains proteins, electrolyte, gases, nutrients, and waste products. Serum refers to plasma minus its clotting factors. Um, you have something called plasma proteins. When you look on the labs, you see albumin, globulin, and clotting factors. These are all plasma proteins. Blood cells make up 45% of blood, And the RBC we know is the job is oxygen transportation. WBC we know it's protection from infection. And thrombocytes, it promotes coagulation, otherwise will bleed to death. Um, RBCs RBCs are flexible cells with very thin membranes, which enables them to pass through tiny passages such as capillaries and to facilitate the diffusion of gases. So RBCs have very thin membranes and they transport oxygen and carbon dioxide. Hemoglobin, in other words, is iron plus protein, and oxygen hemoglobin is equals to oxygen plus iron, which you all have already learned in the first chapter we did. RBCs also helps to maintain acid-base balance. Now, when I was telling you about uh, the RBC has thin membranes. They are made like that, so the gas transport or gas exchange or the perfusion of the RBC or the diffusion of gases via RBC membranes can happen easily. And RBCs are also very flexible. Whichever blood vessel, whichever capillaries they go, they can easily squeeze in and they can travel. So think about the thin capillaries you have in your brain. So these RBCs can very easily travel with those thin capillaries inside the brain and can participate in the tissue perfusion. Um, I don't need to tell you that hemoglobin is very important part of RBC, and it is a complex protein, and it's a molecule, large molecule that functions to bind with oxygen and that functions to bind with oxygen. And as RBCs recirculate through the capillaries surrounding your alveoli within your lungs, oxygen attaches to the ion on the hemoglobin. Oxyhemoglobin, as we know, is responsible for giving arterial blood its bright red appearance. As RBCs flow to body tissues, oxygen detaches from the hemoglobin and diffuses from the capillary into tissue cells. So as RBCs flow to body tissues, oxygen detaches from the hemoglobin and diffuses from the capillary into the tissue cells. Carbon dioxide diffuses from tissue cells back into the capillary. It attaches to the globin portion of hemoglobin and is then transported to the lungs for removal. Hemoglobin also acts as a buffer and plays a role in maintaining acid-base balance. Erythropoiesis, this is the process of RBC production and is regulated by cellular oxygen requirements and general metabolic activities. Um, Erythropoietin is actually a glycoprotein growth factor synthesized and released primarily by the kidney that stimulates the bone marrow to increase erythrocyte production. So you can imagine if something happens to kidney, the erythropoietin will get affected. Normally, the bone marrow releases 3 times 10 to the power 9 RBC per kg of body weight per day. The normal lifespan of RBC or erythrocyte is about 120 days. Several different types of cell types, they evolve when the RBC is maturing, like it's when it's maturation. The reticulocyte is an immature RBC cell that can develop into mature RBCs within 48 hours of release into the circulation. The reticulocyte count, it measures the rate at which new RBCs appear in the circulation. So it is a useful means of evaluating the rate and adequacy of RBC production. So if there is a disorder where the RBC production is getting affected, we want to know the reticulocyte count, which will be helpful in evaluating the rate and adequacy of erythrocyte production. What is hemolysis? Hemolysis is the destruction of RBCs by monocytes and macrophages, which normally occurs in the bone marrow, liver, and spleen. Because one of the components of the RBCs is also bilirubin, hemolysis of these cells results in increased bilirubin to be processed by the body. When hemolysis occurs, normally the liver is able to excrete all bilirubin that is released. So erythropoiesis is influenced by the nutrient availability. And the two things are important is reticulocytes, they mature into RBCs and a good indication of production rates. And hemolysis removes abnormal, defective, damaged, and old RBCs from circulation. Okay, so there are leukocytes. They appear white when they are separated from blood. And like RBCs, leukocytes are made by stem cells in the bone marrow, and they have a variable lifespan. Uh, they can have a lifespan from hours, from few hours to years. There are different types of leukocytes, and each have their own different function. Um, a, leucos, a granulocytic leukocyte is the one which has granules in their cytoplasm and are called uh, and are called polymorphonuclear leukocytes. Agranulocytes or agranulocytic leukocytes are those who don't have no who don't have any granules in their cytoplasm and have only one discrete nucleus. Now, granulocytic leukocytes are neutrophils, eosinophils, basophils, and agranulocytic leukocytes are lymphocytes and monocytes. Neutrophils are the ones which are present in a very high amount uh, in the leukocytes, almost 60 to 70 percentage. Now, if we look in detail, the primary function of the granulocytic is phagocytosis, a process by which WBCs engulfs or ingests any unwanted organism and then digests and kills it. And I don't need to explain you much about phagocytosis. You have been learning it since the time you took your prerequisites for your nursing school. Neutrophil, most common form, with 50 to 70% of all leukocytes being neutrophils. Eosinophils is only 2 to 4%, and basophils is less than 2% of all leukocytes. What does eosinophil do? No, the neutrophil is actually the primary phagocytic cell, and it is very much involved in the acute inflammatory process. Eosinophil has a similar function like neutrophil, but it has little reduced ability for phagocytosis, and it is only 2 to 4% in the whole WBC lot. Basophils, they have chemical mediators such as heparin and histamine, and therefore they are part of allergic and inflammatory reactions. They make up less than 2% of all WBCs. So basophils, they contain chemical mediators such as heparin and histamine, and they are part of allergic and inflammatory reactions. They make up less than 2% of all WBCs. Now we are going to the agranulocytic lymphos, leukocytes. And agranulocytic leukocytes are the ones which do not contain granules, and as I said earlier, and they are only one nucleus, mononuclear. There are two kinds of agranulocytic leukocytes, which are lymphocytes and monocytes. Lymphocytes, they have B cells and T cells, and they form the basis of the cellular and humoral immune response. Most of these lymphocytes are circulating in the blood, and they are also living in lymphoid tissues. NK cells, like natural killer cells, are lymphocytes that do not require any prior exposure to the antigens to kill virus infected cells and activate T cells and phagocytes. So monocytes are the other type of A granular leukocytes, and they account for 4 to 8% of total WBCs. They are also very important phagocytic cells, and they will ingest large masses of matter such as bacteria, dead cells, tissue debris, and old or defective RBCs. And they are only present in blood for a very short time before they migrate into tissues and they become macrophages. These resident macrophages are given special names, and they protect the body from the invading pathogens at these entry points and are more phagocytic. So agranulocytic leukocytes are lymphocytes and monocytes. Lymphocytes, their main role is of the cellular and humoral immune responses. Monocytes, they are uh, very important phagocytal cells that take in or ingest small or large masses of matter such as bacteria, dead cell, tissue debris, or old or defective RBC, and they are in the system for a very short time before they migrate into tissues and become macrophages. We are coming back to the neutrophils. Now, primary function is phagocytosis, and primary phagocytic cells involved. And now, I don't want you to, though it's on the slide, I don't want you to pay much. Just it's good for you to know that they are mature neutrophils which are segmented with multiple segments in its nucleus. I'm not going to go deep in that. Yes, but I do want to say that when there is increased neutrophil levels in the blood, that is a common diagnostic indicator of infection and tissue injury. So that's why we look in the lab sometime how many neutrophils are, how much neutrophils have got affected by any infection or tissue injury. No, I don't want you to go deep in this slide, which is slide 17. Then there is they are showing a normal segmented neutrophil in this picture in slide 18. And then you have 3 bands and 1 mature segmented neutrophil. They're showing you that. It's just good to know. Then when you have an infection, the first kind of leukocytes which will come into action is neutrophils. And increase in percent of bands is called a shift to the left. No, I don't want you to go deep into that, but I want you to know that when somebody has an infection, the first thing in the lab which you can see is the count of neutrophils going high. And they will be high for at least they will be there for at least 2 to 14 days, approximately 2 weeks. In slide 21, they are talking about eosinophils. They are involved in allergic responses, or some skin diseases, or some cancerous disorders, or some in connective tissue disorders. And eosinophils are actually very important in fighting parasitic infections. And so this is the only thing I want you to know about these neutrophils, is that they're only 2 to 4%, 2 to 4% of WBC, part of WBC, and they have a reduced capability of phagocytosis as compared with neutrophils, and they have something called histamine. That's why they are involved in allergic responses, and they are very good to defend against parasitic infections. Eosinophils defend against parasites, and they are found in large numbers in lungs and GI tract. You see the eosinophil on the stained slide. Slide 23 is a picture of eosinophil on the stained slide. And then we talk about basophils. They make up less than 2% of all leukocytes. They have very limited role in phagocytosis. And they have granules which contain heparin, serotonin, and histamine. When they are stimulated by any antigen, basophils will release their granules. So now you see in these pictures there are basophils. So they are normal RBC on slide on right and hypochromic. I don't know why this picture doesn't match. It says leukocytes, and then it says basophils. Okay, just I don't know. This picture is actually not making sense to me, though it was part of your PowerPoint. Just skip this slide 25. Just skip this. Let's go to the next slide. A granul Now we are talking about the type of leukocytes which are A granulocytosis, and within A granulocytes we have two kinds, lymphocytes and monocytes. So let's talk about the lymphocytes. They form the basis of cellular and humoral immune responses, and two subtypes of lymphocytes are B cells and T cells. Monocytes, they are very also important phagocytic cells, and they are the second type of cell to arrive at site of injury. So when somebody has a site injury, the first you will see is neutrophils, which are a lot, and then a second one to arrive will be the monocytes. When monocytes they come into the tissue, they become macrophages. So lymphocyte again. Um, And what are macrophages? Macrophages are monocyte cells that have migrated into tissue. Um, when the macrophages are residing into a certain part of the body, then they have different names. They are called Kupffer cells when they are in liver. They are called osteoclasts when they are in bone, and they are called alveolar macrophages when they are in lung. Um, Thrombocytes, now we are coming to the thrombocytes platelets. We discussed, have been discussing in the class, and we said about the thrombocytes earlier too when we were doing some questions. It initiates the clotting process, it plug openings in the capillary wall, and they play a role in clot shrinkage and retraction. So the primary function of thrombocytes is to initiate the clotting process by producing an initial platelet plug in the early phases of the clotting process. Platelets must be available in sufficient numbers and must be structurally and metabolically sound for blood clotting to occur. So, and platelets have an average lifespan of only um, 10 days, approximately 10 days. Minimum it might go to 8, or higher it can go up to 11, but approximately it has an average lifespan of 10 days. Um, a major factor in healthy functioning blood is iron. Iron is obtained from food and dietary supplements and digested in the upper GI tract, but only about 5 to 10% of ingested iron is absorbed. Most is bound to the heme in blood as hemoglobin and in muscle as myoglobin. The other one third is stored as ferritin and hemosiderin in the bone marrow, spleen, liver, and macrophages. So a major factor in healthy functioning blood is iron. Iron is obtained from food and dietary supplements, and it is digested in the upper GI tract, but only about 5 to 10% of ingested iron is absorbed. Most is bound to the heme in blood as hemoglobin and in muscle as myoglobin. The other one third is stored as ferritin and hemosiderin in the bone marrow, spleen, liver, and macrophages. Now, when the stored iron is not replaced, hemoglobin production will naturally get reduced. Transferrin, something called transferrin, which is synthesized in the liver, it serves as a carrier plasma protein for iron. The degree to which this transferrin will get saturated with iron is a reliable indicator of the iron supply for developing the red blood cells. There is normally very little iron loss except from blood loss, because iron is recycled after macrophages in the liver and spleen phagocytize the old and damaged RBCs, and only about 3% is lost daily in urine, sweat, bile, and epithelial cells in the GI tract. So it's, there's a process which is ongoing. Homeostasis Homeostasis and its four components are important in minimizing the blood loss when various body structures are involved. The arrest of bleeding is important because then there will be a vascular response, there will be a platelet plug formation, development of the fibrin on the platelet plug, and ultimately there will be lysis of the clot. So that maintains the homeostasis. Um, I didn't want to touch homeostasis. No, so whatever I think I will give you in class, the all this about coagulation, some notes about coagulation. What's happening, the coagulation studies and how the platelet plug formation is important. So I don't think I want to skip these slides. Mm, okay. Also slide 41. Okay, now we'll discuss about an organ called spleen. Spleen is located in the upper left quadrant next to kidney, and its functions can be classified as hematopoietic, which is able to produce RBCs during fetal development. It has the four major functions of spleen is hematopoietic, filtration, immunologic, and storage. Hematopoietic function is the one which we are talking here, and it's from the ability to produce RBCs during the fetal development. The filtration function of the spleen is from the ability to remove old and defective RBCs from the circulation by the mononuclear phagocytic systems. So spleen function is that it does filtration. It removes old and damaged RBCs from circulation. It removes hemoglobin from RBCs and returns iron component to the bone marrow for use. It filters out bacteria, especially encapsulated organisms. Um, Spleen is able to uh, catabolize hemoglobin, which is released by hemolysis, and return the iron component of the hemoglobin to the bone marrow for reuse. So spleen is good in catabolizing hemoglobin. Next, slide 44. So we'll discuss a little bit about immunologic function of spleen. And because is from the it has the immunologic function because it has a rich supply of lymphocytes, monocytes, and stored globulins, which play an important role in filtering circulating bacteria, especially encapsulated organisms such as gram-positive cocci. So they have a spleen has a rich supply of lymphocytes, monocytes, and stored immune globulins. At the storage, it stores RBCs, and approximately 30% of total mass of platelets is coming from the storage of spleen. So person, if he has a splenectomy, actually has higher circulating levels of platelets than a person who still has his or her spleen. So the storage function is reflected in the spleen in its ability to store more than 300 ml of blood and about 1/3 of platelets. So a person who has had a splenectomy actually has higher circulating levels of platelets than a person who still has his or her spleen. Lymph system. Yes, blood and lymph both are important for our body, and lymph consists of lymph fluid, lymphatic capillaries, lymphatic ducts, and lymph nodes. Lymph, what is the job of lymph? It enables proteins and fat from the GI tract, and certain hormones are returned to circulatory system. Lymph actually enables proteins and fat from the GI tract and the certain hormones to return to the circulatory system. It returns any excess interstitial fluid to the blood to prevent or reduce edema. So if there is unnecessary fluid floating around in the body, the lymph system will return it to the blood to prevent or reduce edema. Lymph fluid, it is very pale yellow interstitial fluid that has diffused through capillary walls. It moves through a special vasculature, much like circulating blood. So lymph has its own pathway, and it returns excess interstitial fluid, which is important in preventing the development of edema. It's kind of a repetition of the earlier side. Too much interstitial fluid or reduced absorption can lead to lymphedema. May occur as a complication of mastectomy or lumpectomy. Uh, Lymphatic capillaries are thin walled vessels that have an irregular diameter. They are somewhat larger than blood capillaries and do not contain any valves. So they are thin walled vessels with irregular diameter, larger than blood capillaries but without any valves, because blood capillaries have some valves. This is a picture of a lymph drainage system. You can see it's almost all over the body. You can see it's going into the groin area, it's going under your armpit, towards your neck, so it's all over, spread out. And lymphatic capillaries, they unite to form lymphatic vessels that carry all lymph fluid to the right lymphatic duct or the thoracic duct. These larger veins will then drain into subclavian veins in the neck. Lymph nodes are small clumps of lymphatic tissue found in groups along with lymph vessels that filter pathogens and foreign particles that are carried by lymph. Lymph nodes are round, oval, or bean shaped, and they vary in size according to their location. There are over 200 lymph nodes throughout the body, with the greatest predominance in the abdomen, which is surrounding the GI tract. Primary function of lymph nodes is filtration of pathogens and foreign particles carried by lymph fluid. They are situated both superficially and deep. If you do your BSN, or maybe you have done in lab, when you are palpating your lymph nodes, then you say deep lymph nodes, superficial lymph nodes, and you're palpating those nodes. And superficial lymph nodes can be palpated, but evaluation of the deep nodes requires radiologic examination. So, doctors, when they're doing your physical, they palpate your lymph nodes, the superficial ones. Um, the liver functions as a filter. It produces all the coagulants that are essential to homeostasis and blood coagulation. It stores excess iron, and it produces hepcidin, which regulates iron balance. Now, what is hepcidin? Hepcidin is produced by the liver and is a key regulator of iron balance. The synthesis of hepcidin is stimulated by iron overload or inflammation. Hepcidin reduces the release of stored iron from intestines and macrophages. So when the iron is deficient, hepatocytes produce less hepcidin. So I'm not going to test you a lot on hepcidin. If I have to, I will only want to know what's the liver function, what it stores, and it produces hepcidin, which is a key regulator of iron balance. How the hematological system changes can happen in old patients is important. They can be the gradual physiologic process of cell loss and organ atrophy that occurs with aging, and so it can be changes in these. The bone marrow mass and the cellularity will decrease. Bone marrow fat will increase. Although the older adult is still capable of maintaining adequate blood cell levels, the reverse capacity leaves the older adult more vulnerable to possible problems with clotting, oxygen transport, and fighting infection, especially during periods of increased demand. Hemoglobin levels they begin to decrease both in men and women after middle age, with the uh, mostly seen in the older adults. Healthy older patients are not able to produce reticulocytes in response to hemorrhage or hypoxemia, as well as younger adults. So when there is an old patient, they are not able to produce reticulocytes as much as like young person when they are going through hemorrhage or hypoxemia. Platelets functionality can also be reduced. The total WBC count and the differential count is said that it's generally not affected by aging. Uh, but there might be a decrease in a T cell function, which is responsible for some antibody response. So it can happen because of some T cell function decrease. There can be some changes in WBC count, but usually the total WBC count and differentials are not generally affected by aging. So these are some monocytes picture. Next slides you have, these are some neutrophils picture. These are some lymphocytes picture. These are some basophils picture. And the last slide is of eosinophils. So this brings us to the end of this presentation.